and we're talking Verticillium stripe and canola, a European perspective, with Dr. Andreas von Tiedemann, darn it, I forget already, Andreas, but I do have a bit of a biography. I've shortened it up a little bit just because I want it to be mainly about your presentation. So he has a PhD in plant pathology. He was at the Bryce Thompson Institute in Ithaca, New York for a couple of years, and then 25 years at least in Germany, working on understanding environmental and agronomic drivers of crop diseases, including fungal pathogens, the epidemiological and life cycle studies of with novel agricultural pathogens, the interactions of soil-borne pathogens with their hosts, phenotyping of plant diseases, understanding resistance mechanisms in the crop plants to the pathogens, and an integrated management of diseases in a bunch of crops, oilseed rape, wheat, barley, and maize. I'm going to leave it there and just turn it over to him to start focusing on Verticillium stripe based on the experiences in Europe. We have definitely had incidents here in Manitoba especially, and we're looking forward to this presentation. So welcome, Andreas.
Thanks a lot, Tammy, for that introduction. And thanks a lot also to Larissa for managing my participation today, which is a pleasure for me to do. And I think I'm going to share my screen. Is that okay? So yes, I'm happy to give you a short compilation of results we have collected in the last 15 years, I would say, in my oilseed rape pathology group, which is one of my lab groups. And you can see quite a couple of people who have contributed to that kind of research in the last 15 years, PhD students, bachelor, master students, and so on. And I thought I'm collecting and selecting those results which should be of interest for you as canola growers in Canada, so more on a practical perspective. And I will start with looking at the life cycle of this pathogen, how we understand it at the moment. And we do see a monocyclic pathogen, which is obviously living in 4 different types of lifestyles, starting with a biotrophic stage, followed by an endophytic stage, which is quite important and extended during the lifetime of this pathogen within 1 cycle at least. And then it's followed by the necrotrophic stage, which is the first one to become symptomatic. That's what we see when the disease is breaking out on the plant. And this is followed by a dormant phase. I will briefly go through these stages and then fill it up with some of the findings we have made, which might be also of interest for you as a grower. So starting the biotrophic stage, it is quite visible when the fungus starts to infect the roots, as you can see here. I don't know if you can see my pointer. That's good, okay. So it's first producing sort of a superficial hyphal mat on the surface of the roots, and then you can see here nicely how the hyphae are getting attached to the rhizodermis and are starting to penetrate the rhizodermal cells, which is the initiation of infection close to the root hairs. And soon later, if you look more close, you can see as the fungus is also penetrating cortex cells. Although these cells are not responding in any way, they are not showing any necrotic reactions, which indicates that we are looking at a biotrophic interaction, which is confirmed by some biochemical data, which have been produced by a former PhD student in my lab, who was looking at ROS responses, the reactive oxygen species in roots and shoots, and was unable to find any significant responses in this stage of infection. So this indicates quite clearly that we are looking at a biotrophic initial stage of infection, which is, however, followed by a very extended, this is a very short stage, the biotrophic stage, but then it is followed by an elongated, extended endophytic stage. And this is illustrated by these images here. You can see some of the vessels in the shoots which are filled up with mycelium. And the interesting thing is that you would find single vessels which are completely colonized, while the neighboring vessels could be completely free of any colonization. This was the first indication that we have a very linear type of colonization in the vasculature of the plants. And that was also studied more closely by transmission electron microscopy. And there you could see that, in fact, the Brassica napus seems to be able to contain the fungus in single vessels, and it seems to be able to avoid any lateral growth, which you can see in this close up here, where the fungus is attempting to get through the pits, but there is a swelling of the pit membrane, which obviously avoids the transfer of the fungus through the through the side walls of the vessels. So all in all, this means that even under heavy infestation, we do see quite a substantial number of vessels which are remaining functional in terms of water supply. And that for us is one of the reasons that this is the unusual case of a verticillium disease, which is not associated with wilt. That's why we are trying to avoid the term wilt, which is not correct in the case of verticillium, and you correctly announced this disease as verticillium stripe, which was quite interesting when we were looking at the growth of the fungus and the colonization of the fungus in the field, in field-grown winter oilseed rape in roots, hypocotyl, and stems. By measuring the verticillium DNA in these different tissues, was that there was an extremely long period of time in which this infection remained more or less silent. And you can see, starting in September, when the crop was established, we could see some signals with the PCR analysis, and then it became silent over several months, until only in May, the fungus started again to become active and started to really colonize the different plant organs, particularly in the susceptible cultivar, which you can see here, which is ficon, and the resistant one remained on a low level. So we do see for a long while in winter oilseed rape no shoot invasion, and we were not really able to explain that in the beginning, and to understand why we have these long extended period of time in which no symptoms occur, which only occur, as you know, close to maturity stages. But the answer to that phenomenon is probably found when you look at soil temperature. So when you measure the soil temperature at the same time, you would see that you have a particular pattern of soil temperature starting at higher levels in autumn. It's around 15 degrees Celsius in the soil, which obviously enables the fungus to start initial infections. But then the temperature is dropping over winter down to about 5 degrees Celsius, and only in May it gets up again above levels of 11 or more. And we try to test the temperature effect on infection in a climate chamber experiment, which was conducted by a student in my lab, Annette. And in fact, there was a very clear-cut threshold temperature somewhere between 12 and 15 degrees Celsius, which was required for infection of the plants. So there is a threshold temperature in the soil, which might explain the long latency of development of the fungus and of symptoms which you could see in the field. This was also confirmed by experiments in these mini plot soil heating experiments. We established these plots with cables, which would allow us to increase the soil temperature by 1.6 or 3.2 degrees Celsius over the whole season. And we were also able to see a clear temperature effect of soil temperature on verticillium colonization of plants measured as verticillium DNA. So there's an impact of soil temperature on disease. Coming to the next stage, the necrotrophic stage, which might be the most known one, because then you can see that you see these stripes. The stripes are ruptured after some time. Here are some photos of these kind of symptoms, how we find them in our region here, starting with these brown stripes, which are later rupturing the stem epidermis, and then you could see masses of microsclerosia, which are found on the infected stems and also on the stubbles. The interesting thing about verticillium is that you see completely different disease development patterns in greenhouse inoculations and in the field. In the greenhouse, you typically see that the development of the disease is very fast, and it causes a substantial stunting of the plants. And you also see some particular symptoms on leaves. This symptomatology is completely unknown from the field. In the field, you would never see, we never were able to find any stunting effects, even in highly infested plots. but we did see, of course, in inoculated plots, these premature senescence. So this is another result, probably, is different symptomatology in greenhouses and fields have to be taken into account in any studies on verticillium. So what you see in the greenhouse, you must confirm in the field, otherwise it's not a finding which may be of large relevance. And this may also explain why we had really difficult time and difficulties to find significant yield effects of the disease if the yields were really measured under field conditions. So to summarize yield effects under European conditions, we did single plant analysis, and there we could see single plants with a reduced yield about 30 to 60% at moderate or high disease severity levels. But if you're looking at whole plant stands, whole plots, it was very difficult to really see significant differences. And what we found is that there sort of is a tolerance in winter oilseed rape, at least under our conditions, up to a disease incidence of 50%. So Brassica napus seems to be quite tolerant to this disease. Although above those levels, you would see, of course, also a yield decline. What we also analyzed was seed oil content, which was not affected by the disease. So one point which might be interesting for growers and for you guys in Canada is the question whether longisporum is seed transmissible. So why are we asking that question? The question is asked because we have seen that the disease has jumped over to your continent. I think it was in 2014 in Manitoba first. And then 1 year later, as far as I have seen the reports, it was found in several provinces. So that is quite unusual for a disease or for a pathogen like verticillium, because when you look what this pathogen is doing in terms of propagation units, it's producing quite late in the season, mainly on the plant debris. When the plants are decaying far after harvest, you could see these masses of microsclerotia. And this is not a type of disease which you would normally expect to be on seeds. There might be dust in this situation if you are harvesting and you're threshing the crop, which may lay down on the surface of the seeds, but a real seed transmission looks different. So you would normally expect that seed transmission is associated with a systemic growth of the fungus into the seeds, and then it's really established in the seeds. So we were interested to really look whether this is possible. Here's another close-up of the plant residues as we find them in our fields, which are filled up with masses of microsclerotia, which of course is the initial inoculum being reincorporated into the soil. So what we did is first starting with greenhouse experiments, which a Chinese student did in my lab, a PhD student Cheryl, and she did very extensive studies, first with plants which are infected in the greenhouse. As you can see again, you can see the stunting. She collected the seeds and she plated the seeds on agar, and in fact she found the pathogen. So to put that in numbers, she did that with different types of cultivars, with a susceptible cultivar, a moderately susceptible one, and a moderately resistant one. And you could see there was an effect of resistance on the rate of seed infestation, which on the maximum was 13%. So in 13% of the seeds, we were able to plate the pathogen from. So there is theoretical pathway from a root infection to the seeds, no doubt. And you can also see that if you look at DNA contents of verticillium in pods and seeds, where again, we did find signals indicating that the fungus is able to make it from the roots to the seeds. But remember, this is something which does not happen in the field. We don't have this kind of situation in the field, as I mentioned before. And that was confirmed by studies we did. This is another interesting point, is this one. Besides looking at the transmission of the pathogen to the seeds, you always have to look whether the disease is also transmitted. This is quite an important difference to make. And you can see that from the 13% of pathogens transmission to the seeds, only about 2% resulted really in a transmission of the disease, where we could find the disease on the progeny of the plants which have been generated from infected seeds. So it's even under these extreme conditions in the greenhouse, it's a very, very low rate of transmission of disease. And it becomes even more scarce when you look at seeds which are produced in the field. So this was a study in a field experiment close to Gottingen with winter type and spring type oilseed rape. And you can see seed infection was studied again by plating seeds, and there was no single case. We analyzed more than 5,000 seeds here in those two experiments. So it was not a single case where we were able to detect verticillium from seeds of the winter type plots, and there was a very low range, a low percentage of seed infection in the spring type. And also the DNA signals were very, very low, which was close to background noise. And when we used these seeds from that experiment, which by the way was an experiment with extremely strong infestation of the plots of the soil, so high disease pressure actually, we analyzed 480 plants over 6 months, and in no case we were able to find disease on these plants. So the final conclusion, which we also published in Plant Disease, is that from these experiments, there's no indication that the disease can be transmitted by seed produced in the field. The difference between the experiments in greenhouse in terms of seed transmission and in the field are well explained by the different development speed of the disease in the greenhouse and in the field, as I have indicated before. Well, what is the option of management of verticillium? How do we see that? And I think there are two things which can be done. One is cultivar resistance, and the second one might be biocontrol, because biocontrol is a typical strategy to handle soil-borne diseases which are not controllable by fungicides, as you know. So let me look a little bit into those two areas where we did some research, starting with biocontrol, which was, to make a long story short, quite disappointing. We were using a Canadian strain of Microscopia ochracea, which has turned out to be quite effective against a number of important pathogens like apple scab and sclerotinia and so on, from the literature at least. And we did use these strains in vitro, where it performed well. As you can see here, so this is a verticillium colonies in black. When we added Microscleropsis, you would see that verticillium would vanish, and instead the mycoparasite would grow, which is white. So whenever you see a white colony, it was successful. The black colony is verticillium. So in vitro, we saw quite some effect, about 90–95% efficiency. But the more we went into unsterile conditions, from the lab to the greenhouse, to the mini plot field experiment, to the large field experiment, you could see that we would completely lose that effect. So finally, in the field, there was absolutely no effect, no biocontrol effect by Microscleropsis. So we had to give up that. And another student tried to use some other biocontrol agents like Verticillium t-corpus, Verticillium isaki. We also used Piriformospora indica and Rhizobium, and all these attempts were unsuccessful. Much more successful were our studies in improving cultivar resistance, which is a long-standing collaboration with German breeding companies and some colleagues from the breeding department in the University of Gießen. I will briefly show you some results of these studies. The starting was already promising, as we were testing a number of lines, also parental lines of Brassica napus, which is cabbage and turnip rape, I think is called in English, Brassica rapa and Brassica oleracea. So we tested these lines, and we immediately found quite a variability in responses to infection, which is the starting point of any type of breeding. You need variation, of course. And it turned out that the cabbage lines, which are in the blue bars here, are having a better resistance in general than the green bars, which are representing the Brassica rapa parental lines. So from these findings, it appeared that if there is any quantitative resistance, it should be mostly localized in the Brassica oleracea genome. Which turned out to be true. You can see that there can be substantial differences. This Swedish line, for example, has strong resistance. You can see this is infected here with nearly no symptoms, while others are really reacting in the expected way by stunting and so on. So there is quite a good basis for breeding, and over time we also saw in the different sets of lines we were testing for the breeders that there was a continuous decrease in the mean level of disease, and meaning an increase in resistance. And the colleagues from Gießen did a QTL analysis also to see where the resistance is located, and the results were of course interesting to develop markers for marker-assisted selection. And they found 3 loci on C1, C2, and C5 chromosomes of Brassica oleracea, which explained about 30% of variability and resistance. And this was well in agreement with our findings that the Brassica oleracea lines were more resistant in general as compared to Brassica rapa. The resistance is interesting. It's a vascular resistant. It's not a root penetration resistance. So what happens actually is when the fungus is so even in a let's say in a disease situation, or in a resistant situation, the fungus would be able to penetrate the roots, but then it is stopped actually in the hypocotyl tissue. And when you look closely what happens there, you can see that there's a substantial accumulation of phenolic substances in the vessels. Here is the Swedish line down here, and this is a susceptible line Falcon. And we saw substantial differences in the phenolic responses of these two contrasting lines, which was indicating that the phenolic responses are part of the resistance mechanisms, which are finally not really clear, but there seems to be an involvement of phenolics at least. So it is a penetration resistance which is avoiding the invasion of the shoot and is containing the fungus more or less in the roots, which avoids the most of the damage. In the last years, we were also interested in population genetic studies on Verticillium lineages. As you may have seen, there was a paper by D. Potter, and before that, there was another paper by Inder Bitzin from UC Davis, indicating that there probably are 3 different lineages of Verticillium longisporum, designated as A1D1, A1D2, and A1D3. So these are 3 different hybrids. As you know, Verticillium longisporum is a hybrid fungus, nearly diploid fungus, composed by 2 haploid genomes. And these 3 lineages actually are 3 different types of the pathogen based on the hybridization of 4 different parental genomes. And since that finding, we wanted to see how this translates into aggressiveness or pathogenicity of these different lineages, and also we were interested in a monitoring to see what the populations consist of, which we find outside in the field. And so Marta, Spanish PhD student in my lab, is just finishing her PhD right now, did this multiplex PCR, which is allowing to differentiate between A1D1, A1D2, and A1D3 and Verticillium by the different banding patterns. And she used that to differentiate the strains in our collection. First of all, we were interested to see whether the D2 strains were also pathogenic, and it turned out they are not. You can see 3 examples here, and the plant is shown here that A1D1 turned out to be not pathogenic on Brassica napus. You may remember that, which I show here, that A1D2 was mainly found on horseradish, in a few cases also on oilseed rape, but it obviously doesn't seem to be aggressive on oilseed rape, indicated by very low net AUDPC, which is indicating infection, and no stunting. So the plant height is unaffected. while a D1 strain, which is the VL43 here, which is the D1 strain, is aggressive and is causing disease and stunting the plant. So that was interesting, and she was also interested to see whether there is an interaction between D2 strains and D1 strains. And interestingly, she found that D2 even has a biocontrol activity in terms of reducing colonization of oilseed rape with aggressive strain, which you can see here. This is a plant which has been pre-inoculated with a D2 strain and then inoculated with aggressive strain D1, and you can see there's no disease. Here's the control, and here's the pure inoculation with the D1 strain, which is causing disease. So both, especially in simultaneous inoculation with the two strains or with the two lineages of Verticillium, you would see a substantial reduction in colonization of the plants. So this is a side effect of these studies on lineages, but the main question we were asking is how are the Verticillium populations in the field composed of in terms of lineages. And so we collected a large number of isolates over several years from many locations in several countries, including Canada also. There were 2 places from Canada where we got a total of 7 isolates. I think there's both in Manitoba, I think. I guess so. Should be both in Manitoba. I guess these are strains from your place. And Marta did a GBS analysis with a large number of these strains. And just to give you a short insight into what she found out is that the first thing which I should mention is that all the isolates in our collection turned out to be A1D1. So whatever we found is from one lineage, and these seem to be the aggressive type, which is found in symptomatic plants. We have to consider that all the isolates, of course, were isolated from symptomatic plants, and so there A1D1 seems to be prevalent. But when we are dissecting this population of A1D1, which was comprising of 81 isolates from Europe and 6 from Canada, we found an interesting grouping. So there were 3 groups, one big group here, the second group here, and the third cluster here. And when we were trying to assign that to geographic origin, we found an interesting pattern in that all the Canadian isolates came from one cluster, while the English isolates came from a second cluster, and the German, French isolates and from some other countries with traditional cultivation of oilseed rape were stretching out over all the clusters. So we're present in all the clusters, which is indicating probably that the Canadian isolates are deriving from a more recent introduction of some single strains, and so we don't see that genetic breadth, this quite narrow genetic range here, and the same might be true with England, which is an island, as you know, and might also be affected only by a single and isolated introduction. Marta also looked at pathogenicity of these A1D1 isolates, and just want to give you a brief impression of what she found out. This is again a study of the net AUDPC, so the area under the disease progress curve with 2 different cultivars. And the message from here is that we see quite a diversity of aggressiveness levels from all these different isolates. They are all D1 isolates, as I said, and there is not a systematic difference between the 3 groups represented by the 3 genomic clusters. And for us, it seems that there is a diversity also and a variability in aggressiveness, and what we may expect from long-term growing of canola is probably a selection of those ones which are more aggressive. That is one thing which we should keep in mind. With that, I come to my conclusions. And again, I would like to highlight that we are talking about a disease which is not a wilting disease. It's sort of a premature ripening disease. We do have a pathogen specialized on Brassicaceae, and this has been found in other studies before. The yield effects on winter oilseed rape are moderate, and we feel that this is due to the extended latency of symptom development, which mainly occurs during winter time when temperatures in the soil are not sufficient to for the fungus to readily propagate and grow in the plants, which means that early sown winter oilseed rape, which means sown more in the warmer part of the season, or spring sown canola, as you are growing in your country, may be at higher risk, because soil temperature thresholds of 12 degrees Celsius might be then the relevant factor deciding whether you get infection or not. We have done other studies in which we found, which I haven't shown you by data in this presentation, that humidity also plays an important role. There's no fungicide control, as you may know. We did some experiments with fungicides, but we never were able to have substantial effects. So fungicides are effective in vitro. So the fungus is not insensitive to the current fungicides, but you don't get enough fungicides into the vessels where the fungus is. So there's no control. Quantitative resistance is available, especially deriving from the cabbage C genome. We did some more resistance studies, which I skipped in order to keep the presentation short. And phenols are obviously involved. Also, salicylic acid has been found to be part of the machinery of basal and cultivar-related resistance. More interestingly, maybe, is that among the three lineages which have been identified so far, D1 and D3, I was not talking about D3, but D3 is aggressive also on oilseed rape, while D2 obviously is not. It may be having an activity as a potential biocontrol agent. And in all our monitorings, as I indicated, D1 was the prevalent and the only one we found. And about seed transmission, again, I want to emphasize that it seems not to be very likely that we would get seed transmission by seeds produced in the field based on the results we got from our experiments. With that, I thank you for your attention. I'm open for questions. I would like also to acknowledge all the people who have contributed and cooperated with us during the last years, and also for the financial support by different ministries and funding agencies. And with that nice slide at the end, I leave you, and I'm happy to take your question. Thank you.
Perfect. Thank you, Andreas. So before we get to questions, and we do have one already, we're just going to get Justine to speak on the Canadian perspective. So Justine, for those who don't know, is an agronomist with the Canola Council of Canada, although I think you'd have to live under a rock to not know who she is. So a bit of a cowgirl, but also wrangling with some canola diseases. She's part of the crop production and innovation team with the Canola Council, covers off Western Manitoba and Southeastern Saskatchewan. Her specialty with the council is blackleg and verticillium management. She leads the Canadian Blackleg Steering Group, and she's also, in her spare time, a part-time master's student at the University of Manitoba in plant science. And so I'm just going to turn it over to Justine to talk about the Canadian perspective before we launch into a short Q&A.
Awesome. Thanks, Tammy. Can you hear me and see my slides? Okay, perfect.
I sure can.
Wanted to validate that before I continued on. So this has been great. It's been such a privilege to have Andreas here today to speak with us and really share his expertise of what we know about this disease, mainly because it's something new that we're dealing with here in Canada, right? This is a brand new disease. It was first found here in 2014. Of course, in Manitoba, we sure like our canola diseases. But that was the very first case that was detected here in Canada and in canola. So that was submitted through CFIA, so our Canadian Food Inspection Agency. They discovered that. So that was through DNA detection. And then from there, they completed a really comprehensive survey the following year. So in 2015, they went all across Canada, and they ended up finding it in 6 provinces. So we know it's here, and we know it's widespread. A few other little things just to kind of bring us all up to speed, and Andreas spoke to this several times. There's been a name change. When we first found the Longosporum species here in Canada, it was being referred to as causing Verticillium wilt. So we really are trying to change the industry and go with this Verticillium stripe. I kind of play the cop on that perspective and trying to get our industry to stay on board and to be talking about Verticillium stripe to not confuse producers. So really that's where we were kind of at with Verticillium. Wasn't a really big issue. We'd find a few cases here. We were slowly starting to learn what it would look like. Really untimely phone call. In 2018 and 19, wait a minute. Okay, sorry, there we go. So in 2018 and 19, we had delayed harvests and we were really starting to see the symptoms. And then we bring ourselves up to 2020, the year of all sorts of unfortunate events. We saw really severe symptomology across Manitoba. Right, all the way from Red River Valley into Swan River Valley, we were able to find this, producers, agronomists, lots of reports coming forward. So I will flip over this quickly. We already know what the species we're dealing with. The one reason why I do mention Verticillium longisporum for our potato growers here and our agronomists that work with potatoes, we are quite familiar and work with Verticillium dahlia. So there's a difference there in species. So just to kind of put our Canadian perspective on this, right, we don't know a lot about this disease. We've got research underway. We're just at the point now as agronomists and as producers to identify this disease in field. So like I said, every year we kind of learn a little bit more about it and what we're looking for. One of the really prevalent symptoms that we see in the field is the stem senescing. And typically we are seeing this unilaterally, so only on half of the stem going upwards. This year in some of the fields I was in, you were able to see it up into the side branches and all the way up into the pods. And what else we see is a lot of almost constriction where that plant seems to have kind of dried right down. It becomes very fragile on that one side. And this is another thing not to be then confused with Fusarium wilt, which is a disease that we dealt with about 20 years ago. And hopefully it stays down low. But anyways, that's for another day. One of the other really characteristic symptoms that we are looking for, signs of this disease, is the actual Microscopia. So typically with my field experience, we were finding it up in the stems. So right, seeing that shredding of the outer layer, that outer stem wall to reveal the Microscopia below. The cross-section cut, which I was seeing this year a lot more, is within the root tissue. So going in to do your black leg ratings, pulling up those roots to have a look for club root, anything like that. When you actually cut through that root tissue, you were able to see the Microscopia forming within that root tissue. So seeing this gray speckling across the entire cross section was another way to actually find and identify the disease. Here just saying late fall, typically in the past few years, it was late fall, post harvest, you'd go out and if you're out soil sampling, you'd be able to find it. This year, just the environmental conditions were really conclusive for disease development. You were able to find these symptoms very early, late August, even mid-August, we were seeing a lot of that stem senescing. But like I said, in the past, we have been looking later in the season and really looking for that shredding of the stem wall to reveal the Microscopia below. So just as an update of where we are at, this year the provincial governments all completed their canola disease surveys and they were looking for Verticillium stripe this year. Alberta continues to have a very low prevalence. So I think actually it was only 4 fields out of the 300 to 400 fields that they survey. And they were kind of isolated to one county there, but same, they were able to find it, but only a few plants. Same kind of situation in Saskatchewan. No samples to date were submitted through their disease survey. Some were submitted to their diagnostics lab early on, but they were still waiting to get DNA verification through there. Then we come to Manitoba, the province of Verticillium. So this year, same, we have been surveying, so Manitoba Agriculture through their canola disease survey. We have been looking for this the last few years. It's on one of the diseases that is listed. This year, the rough estimates were that over 30% of the fields that were surveyed in the canola disease survey were showing symptoms for the disease. So we're still trying to finalize that, they're still trying to finalize that number and just to verify that through the lab. Producers had the option and agronomists had the option to submit samples throughout the growing season. And this year there was a huge influx of just overall samples submitted to labs. The PSI lab to date was receiving upwards of over 170 samples that were all confirmed with Verticillium longisporum being present. So that's just like just a rough guess, a rough estimate of where we are at. So we are seeing this and we are seeing it very widespread across Manitoba. Lots of confusion. We say, you know what, we've probably been dealing with this particular disease for a longer period of time. It was just going, you know, undiagnosed. Very, very similar symptoms, and especially when we go in to do a typical canola disease survey, so around that 60% seed color change, we just weren't seeing the really evident signs of this disease. So the Microscopia were not showing themselves. This was probably confused with blackleg, right? Seeing this discoloration in the stem. This is also very characteristic of Alternaria and Fusarium, kind of seeing that starburst pattern across that cross section in the root tissue. So same, it's probably been here and just misdiagnosed. Same, you know, and we see it, of course, on plants together with other pathogens. So here's a really great example of seeing Verticillium and blackleg in the field on the exact same piece of stubble. We've probably got a Leptosphaeria biglobosa causing the blackleg pycnidia on the stem, where if you peel back that outer stem layer, that's where then the Microscopia of the Verticillium longisporum was then evident. Very same thing then with Sclerotinia. Right, and I think this is at the typical canola disease surveying time where there was confusion. You were seeing the shredding of the stems and stalks, and not, of course, then seeing the Microscopia for Sclerotinia, where you are seeing it with Verticillium. So you can see the color difference there. Just quickly to highlight, here in Canada, like I said, right, 2014 was the first year we figured out that this was here. In 2018, there was a few projects established through our Canadian Agricultural Partnership Program. So this is looped in with the overall canola cluster. But we do have one project that's being led by Dr. Fernando at the University of Manitoba. So really just kind of looking at the ins and outs of this pathogen and disease here in Canada. And the second large project there is then a genetics and genomics project with Dr. Hossein Borhan at Ag Canada, Saskatoon. There's one smaller project as well that the canola grower organizations are funding, and it's just an overall Verticillium stripe management, and that's where the yield loss study is being conducted, and that's at the University of Alberta in Shofang Wang's lab there. So we're getting there. We've got a foundation of work coming out, and we're slowly taking information from that and being able to apply it here. But really, we have been relying on what the work that Andreas and his lab has been able to do and share with us, and to make some of these decisions so far. Some really quick takeaways. Saying we didn't get into management yet, right? We're still a little bit away from that. With this being a soil-borne fungus, the real big concern here is the risk of spread and moving within our soil and within our landscapes here. We've got limited management practices right to date, but one positive thing is it's not a regulated pathogen here. So there's not a concern that way that it's going to be constricted. So we've got a lot of things to move forward with, but I will wrap this up so we can get some questions, because I know for even myself, I've got questions for Andreas today on Verticillium longisporum. So with that, thanks. I will talk about testing later.
Thanks, Justine. Really appreciate it. So we are at 1:45, and theoretically Dan should start, but I talked too long in the intro, so I'm going to give a couple minutes for questions. I think the one that's consistently coming across is the impact on yield and why it's impacting on yield. We're seeing high levels of incidence, but not necessarily translating to yield. And if we are seeing a yield impact, is it a vascular contribution, or is it because of wind and weather after the fact with just a bad stem then? Like, what's going on there? So Andreas, I'd love your perspective, and Justine as well.
Well, I think there's a big difference is that you are growing spring type canola, which is having a shorter vegetation period. And as I said, from all the setting in this kind of production of canola, there seems to be a higher risk of infection. And I could imagine without creating fear with you is that on the long run there might be higher disease levels, and these ones can also translate into higher levels of yield losses, maybe as we do see in winter oilseed rape production in Europe. As you know, we are not growing spring type canola normally. So we did one experiment in Gottingen also with spring type canola, and we did see also a faster development. And this is related to the shorter vegetation period plus the higher temperature in the soil, which the crop is exposed to. So my question would be in terms of the differences which you found between the provinces, between Saskatchewan, Manitoba, and Alberta. Do you know anything about soil temperatures? Is there a gradient of soil temperatures when you grow your crops there, which would indicate different levels of disease also?
That's where I was kind of going with it as well. I was looking at a soil temperature component. And, you know, if it did originate in the Winnipeg area, we are slowly spreading it across the prairies. And it's just becoming more and more prevalent. And that's probably why we're seeing limited cases in Alberta and Saskatchewan to date. As for the yield perspective too, I look at the last, 5 or 6 years, we've really moved into a dry cycle. And I think that's why we're starting to see the symptoms of the disease a lot more evident in the field. That being said, and, understanding now the how this pathogen moves in the stem and being taken up and still able to take up moisture where it's not infected. I think that's why we're probably seeing a lot of plants infected, but not resulting in that yield loss. They are still getting, enough moisture uptake to make it through. So like I said, it's something we are looking at here in Canada as well, to look at our on our hybrids, what the impact would be from this pathogen.
Okay, Justine, I'm going to offer you the option of one very short question to Andreas, and then we'll have to wrap it up.
Oh gosh, there's way too many for that. I guess, Andreas, I know you had highlighted a few different management practices.
Minister of Infrastructure?
Just keep talking, Justine. I'm sure.
Okay, sorry, I hear things. Okay. I guess, what is the recommended practice or management practice that growers are using over in Europe? Are they relying on cultivar resistance?
That's the main strategy now. So we have quite some activities in the breeding companies based on the markers which we found to select for more resistant varieties, and this is underway. And I'm expecting in the next few years that we will see these cultivars on the market also. And with that, I think we can at least keep the disease levels on a disease on a level which is not affecting yield anymore. But we don't know what happens on the pathogen side. As I said, we see differences in aggressiveness, and I'm, as a plant pathologist from many years of research, I'm expecting that we are selecting for more aggressive strains also. So this is an arms race which you always see between improvement of cultivar resistance and aggressiveness or pathogenicity on the pathogen side. This will also happen with Verticillium, I guess.